Okay, so welcome again to the welcome back to the symposium. So my name is Dr. Narinda Ahmad. I'm from the Faculty of Veterinary Medicine, University Putra Malaysia, and I will be sharing session 7 with the theme of zoonosis and raptor medicine. So we have seen just now, Prof. Jalila had nicely give a great insight in terms of the interdependence of research and conservation efforts, between different expertise from various fields. So not only is One Health important to be considered when you talk about conservation efforts, but also when you are trying to work together, you would have to consider and in terms of working across different sectors. So I would like to invite our first speaker for today, coming in from Indonesia. So I would like to invite Dr. Andreas Bangphra from the Laboratory of Veterinary Anatomic Pathology, Faculty of Veterinary Medicine, Universitas Brawijaya. So because, so you will come in first and then we'll proceed with Dr. Surawit's presentation later when he comes in. So Dr. Andreas, are you ready?
Yes, I'm ready here.
Okay, yeah, the floor is yours. You've got 20 minutes.
Okay, I will try to share my screen first.
Okay, sure.
Okay, can you see my screen and hear my voice clearly?
Yes, great.
Okay, great. Try to put it in full screen mode. Okay, thank you very much for the occasion given to me, and good afternoon again, and good morning and good evening for those are in the different time zones. And my name is Andreas Bangphra, and here I'm talking on behalf of the Wildlife Rescue Center Jogja, and this is the field project of Yogyakarta Nature Conservation Foundation. And I'm also a junior teaching staff from the Faculty of Veterinary Medicine, Universitas Brawijaya, Indonesia. And this time, actually, I replaced my colleague here that is the primary researcher, Dr. Wari Pulung-Nugrahani, but she's out of range of the internet, so that she cannot join our symposium today. And then this is the case report, actually, the simple case reports that we keep from 2019. And as you've seen that in this screen, it is actually a brain of the Changeable Hawk-Eagle. And we trim it in the what we call the breadloaf pattern. So we trim it layer by layer. And something bizarre over there is this one. You can see this, the protrusion or the mass that coming from the left hemisphere, and also we found 2 cysts, the brain cysts, and we'll talk about it and focus in this pathogenesis for this presentation. So that this is the cerebral cyst in a sudden death Changeable Hawk-Eagle. And a brief about our center. So actually, I work with this center in Yogyakarta. So we are located in Yogyakarta and covering the working region in the Central Java. We are doing rescue, rehabilitation activity of the various animals from the reptiles to mammals, and thirdly attempting to be part of research. And we are affiliating to several universities in Indonesia, helping us performing the research in some animals. And if you are interested in our project, you may check our website and see what we are doing and what is going on at the moment there. And go back to the story about this eagle. So this is an adult Changeable Hawk-Eagle, Nisaetus cirrhatus. Previously, it was known as Nisaetus cirrhatus, and it was found dead in 2019. So it was 2 years ago, and we kept this information, and we just released today. And as you see in the picture, the body just like there in the floor of the cage. After we give them the second feeding, so we usually give the bird in our center twice a day, the morning feeding and also the afternoon feeding. And just after the afternoon feeding, the keeper said that the bird was dead or deceased. And information retrieved from our keeper shows that this eagle is normally catching the lizard in the morning feeding time with no suspicious behavioral disorder or gross physical abnormality. And fortunately, a day before we found it dead, we performed the physical examination, and the handling and restraint procedure were done by our keeper manually using the ABA method. We just immobilized the eagle using the towels and wrapping in the body just to immobilize them so that we can examine everything easily. And we found that the heart rate was 140 per minute and the respiration rate was 40 times per minute. The body temperature is a bit higher, I thought, 36.1 degrees Celsius at that time. And however, all organ systems were apparently in normal condition except the pectoral muscle. But it is usually happening in the bird that was kept in the small cage. So they can do the excessive exercise to maintain the muscle so that the concave muscle and also the atrophied musculature of the pectoral is quite normal in the bird that is kept in the small cage. And then we did the necropsy. Oh, sorry. Previously, we also took the blood sample as well. And you can see that here, the blood reference interval of the Changeable Hawk-Eagle is not yet available in Indonesia. So that we refer to the Javan Hawk-Eagle here. You can see in the second column here, this is just Javan Hawk-Eagle because of the closest relative of this bird. And then to be noted, this came from the previous research and only participated by 4 Javan Hawk-Eagle, so that the result is in the low accuracy and maybe high statistical error. So we just put it as the comparator as well. However, it is still the best reference we could obtain considering the data deprivation of our endemic eagles in Indonesia. And you can see that it can be noted that the most of red blood cell parameters and indices are elevating. So in this part, so you can see in the total erythrocytes, the HB, MCH, MCHC, unless the MCH shows a slight decline compared to the reference. However, the increase of the red blood cell is maybe associated with the dehydration condition. So that can be in the fake polycythemia, I can say. And then the plasma protein is slightly rising, whereas no reference of fibrinogen is available at the moment. And all white blood cell parameters are within the range interval. And yes, like I have mentioned previously, because of species difference, it may not statistically comparable as a basis to draw a conclusion, so that we just rule this hematologic result out from this evidence list. And then, yes, this is the necropsy, the result of our necropsy. A day after we found the eagle's dead, we previously preserved the body, the carcass, in our freezer and thawed it before the necropsy. And once we open the upper digestive tract, you can see that the whole body of the second lizard we gave to this bird is still intact. So that it was actually unable to chop it. Usually they chop the body of the lizard and eat the part by part, but this one is a bit different. So this fact eventually led us to the assumption that the eagle died just after swallowing the whole body of the second lizard. And in addition, it is also a strange behavior that the eagle did swallow the intact lizard's body instead of tearing it apart first. Since the excessive load of esophagus may compress the trachea and eventually blocks the windpipe or airway, we thought that suffocation due to the tracheal compression is listed in our differential diagnosis list. However, we could not ensure that it will cause the sudden death in this event. And if you see in the upper picture, there are the 2 lobes of the liver, and there is an approximately 2 millimeters in diameter, moderately demarcated and yellow focal, flat worm-around lesion in the visceral surface of left liver lobe. And we also see the flat yellow pinpoint lesion in the liver is very typical to hepatocyte necrosis or early development of focal necrotizing hepatitis. And yes, it needs to be proved by histopathology examination to support this diagnosis. However, we thought that it's not significant to the sudden death event, so that we also rule them out. Then the most distinctive lesions are in this picture. You can see the empty cavity resemble a cyst located in the parenchyma of the right cerebral lobes and the tumor-like nodules. This one, and it is actually a round mass, but we accidentally cut it through, so that there is the remain of this part, and this part is still attached from the former tissue. And there are actually two visible cyst-like lesions measured approximately 1 by 3 millimeters and less than 1 millimeters in diameter, respectively, in the first quadrant of the cerebrum, so in this part. And it is elongated from this part until that part. And you can see that small, I don't know whether you can see that one, that is small cyst as well in the roof top of this cerebrum. And then unfortunately, the cyst content is possibly washed out due to the histotechnic processing. And this is the histologic examination result. You can see that there is no hypercellularity, and it confirmed that this is the cyst. This is actually the cyst, and it previously was filled by the something like fluid or maybe the colloid, and then it was washed out. And this is the histopathologic photograph of the mass. So you can see that there's no hypercellularity indicating the tumorous or maybe a neoplastic lesion. So there's not a cancerous proliferation. So we can rule out the neoplastic event in this part. And this is also the prominent lesion we can find at that time. So this is actually in the vein. So the vein is quite dilated in this part, and we could not say that it will associated with the sudden death event. So we draw the presumptive pathogenesis here that the key lesion is possibly located in the brain. So you can see that the tumor-like nodule and the former cyst cavity is quite prominent from this case, and the tumorous mass always compresses the adjacent tissue. This is very classic in this case of brain tumor, and it increases the intracranial pressure, which alters the vascular permeability and pressure, if that is the neoplastic mass. But we also have the second diagnosis list as well. So talking about the neoplastic event, the cancerous tissue also creates the expansive weak vasculature arrangement, which is prone to endothelial destruction and eventually hemorrhage, of course. And if the mass is located in the pivotal center of the consciousness or other important organ system that regulate the physiology of the birth, so it can be defective and impactful to the sudden death. So maybe there is the plausible event of this sudden death in Changeable Hawk-Eagle. And the origin of the cavity is still poorly understood. However, considering the fact that the intercerebral mass may develop edema, localized interstitial fluid accumulation may initiate a cyst formation. And other intercerebral cysts have reported in sudden death case in human as well, but the type of the content is a bit different, because in human we found it as the colloid cyst, so it's a bit a turbid one, the highly concentrated protein inside. But yes, in fact, that in this case we could not find the content due to the preservative and also the washing out event of the histotechnic procedure. So the lesson learned and also the recommendation of this case is that due to the deficient information of reference, the complete and thorough necropsy is recommended for every wild animal disease in our center, because we always did this one, the thorough necropsy, because we would like to see whether there is the variation of the anatomical appearance, or maybe there is something new about the organ in every species we kept in our center. And then some findings might be enigmatic, like this event and this case, but comparative pathology may help for disease pathway deduction. And at last, I would like to acknowledge some parties that help us on doing this research. The first is Ministry of Environment and Forestry of Republic Indonesia for permitting us performing this study. especially in this necropsy, and our counterparts from university. We have Faculty of Veterinary Medicine, Universitas Brawijaya and Universitas Gadjah Mada as well, for assisting us processing the tissue specimens and analysis. And if you are wondering about us, you may visit our social medias. So it's about promotion. And as presented on the screen, and if you are willing to collaborate with us with some research, don't be hesitant to contact us. And our research leader will talk more about this opportunity in the next presentation. And thank you very much. I will give back the time to Dr. Indah Ahmad.
Thank you very much, Dr. Andreas. So that was an interesting case presented there. Do we have any questions? Because we still have time for questions. You can type in the chat box. Okay, questions? All right, if there's none, then just one question from me. So from the two potential causes that you presented just now, so in terms of feeding in large size feeding, so is that possible to happen when you feed? So for example, the bird was given lizard, right? So I mean, was it like given in bulk or how? Because yeah, I mean, I was just trying to imagining the cause. Yeah. So yeah.
Okay. So yes, that's maybe that will be the cause of sudden death as well. But we found the body of the lizard is positioned under the esophageal, so it has been already in the proventriculus. So the suffocating of the tracheal is less possible for this case. But if you ask about how big it is, maybe just like two of my fingers. But usually we give the same size of the lizard for every eagle in our center. That is why we thought that it's not a problem, not the big problem for the eagles, but the behavior that is in our concern, because usually they chop one by one, like swallowing the whole body of the lizard. That's the weight one in this case. Hopefully that answers your question.
Yes, yes. Thank you very much, Dr. Andres. So if we do not have any other question, so we shall move on to the next presenter. So I would like to invite Dr. Irhamna Putri Rahmawati from the Wildlife Rescue Center, Yayasan Konservasi Alam Yogyakarta, to present her paper entitled Hematologic Profile and Value Comparison of Changeable Hawk-Eagle with Pododermatitis and Ectoparasites Infestation. So I would like to welcome Dr. Irhamna.
Yeah, can you see my screen already?
Yes.
All right, thank you very much for giving me again the opportunity to present our work here. It's yeah, I'm very happy to be able to present this to you all guys. So I apologize if later on I will probably just turn off my video because I have a little bit issue with the signal here. But yeah, so thank you. I will begin my presentation here with the title of Hematologic Profile and Value Comparison for Changeable Hawk-Eagles with pododermatitis and ectoparasites infestation. So the background of our study here is actually because this species, the Changeable Hawk-Eagle, are abundantly rescued and yeah, seen in many ex situ facilities here in Indonesia, but at the same time there is no hematology references ever reported for this species. So we thought to fill the knowledge gap about the wildlife medicine, especially to help improving the health management of this species by providing a qualitative as well as the quantitative hematologic reference for this species. So, well, our background for this research is actually first to actually establish the CBC's reference value. So we would like to establish the reference value for several blood parameters such as the PCV and then the hemoglobin and total protein plasma, as well as the number of red blood cells as well as the white blood cells of this species. And the actual method was first is to select how many and which individuals we will include in this study, and we decided to take around 33 individuals from 3 different conservation institutions around Yogyakarta. The first is including our site, the Wildlife Rescue Center Jogja, and then we also took several samples from the Gembira Local Zoo, that's the local zoo near our site, as well as from the government site of Rehabilitation Center. And then we, as the first type, obviously, we will divide the birds into the groups. The first would be the healthy group, and then second group would be the diseased or the unhealthy groups. And for the inclusion criteria of the sample, we exclude the blood samples which already underwent the hemolysis process. And then we will test the blood of both groups following the standard methods. And then after the results came out, we would analyze the results all using the statistic method. Here we use the R statistic software. And then to establish the hematological reference interval, here we underwent several statistical methods, including the outlier elimination method. And then secondly, we did the normality test. And then third, we developed the reference interval calculation. And then because of the things happened in the field that made, actually we didn't meet the minimum requirement to establish the reference interval value, we then do another analysis on the subgroup comparison of the healthy and diseased bird, which I will explain to you later on. And so that's the method. So here is the results. First, I will talk about the result of the reference interval. And then second, I will talk about the subgroup comparison. That's another analysis we did. So first of all, as I said previously, and that basically we divided the birds into two populations, the healthy and unhealthy birds. We only choose the healthy birds to establish the reference interval following the standard procedure developed by the Association of Veterinary Pathology. And then obviously to determine which individuals are belong to healthy and which are those unhealthy, we first did the general checkup which included the fecal and skin check for possible endoparasite and ectoparasite. And we also did the antibody screening against the two most prevalent diseases in our area. Those are the avian influenza and also the Newcastle disease. And we also did the clinical body examination for each individuals. And for the healthy individuals, of course, those that are free from parasites, both endo or ectoparasites infestations, and those that had less than 6 of reactive antibody against avian influenza and Newcastle disease, and those that are free from any physical injuries or trauma. So eventually, we only obtained from 33 individuals included in this study, we only obtained 14 healthy individuals. And the rest of them, which is 19, were unhealthy. So those are really not what we actually expected before, because the actual minimum requirement to establish the reference interval was at least 20 individuals, but we only obtained 14. But here we still tested and analyze statistically. We still try to still stick into the method. And here, as I said previously in the method, that we tested using the outlier elimination first to eliminate the data that are not ideal to further analysis. So basically, we eliminated the data that may obscure the result of the study afterwards. And as you can see in this histogram pictures, yeah, it's very unfortunate that apparently we failed to obtain a good and representative data required for establishing the reference interval. Yeah, obviously this might happen because we only had 14 healthy individuals anyway. And but yeah, we still went through the process to calculate the reference interval, and here is the result for the red blood cell, white blood cell, PCV, total protein plasma, as well as the hemoglobin. And that's the result from the reference interval. And then secondly, we thought of other analysis that that we can do using this data, and we decided to conduct the subgroup comparison test to follow up the result of the general check in unhealthy birds. Yeah, and then here from the unhealthy birds, we divided them again into 3 groups. The first group would be those that the birds with ectoparasite infestation, which was accounted for 11 individuals, and then 8 birds with bumblefoot and 6 birds who were contracted with both the ectoparasite infection as well as the bumblefoot or the pododermatitis. And from this, we analyzed each blood parameters result, and these are the results of the hematologic counts. And this is shown in a box plot and whisker diagram. And this type of diagram is actually very useful to search information about how the data were normally distributed or not. So visually, there is no outliers or there is no extreme number seen in the red blood cell statistical analysis here, but there are several outliers or extreme numbers on the other blood parameters, such as the white blood cell. Here we can see in the dots here, as well as here there is 2 outliers and 1 outliers in the PCV and 2 outliers on the total protein plasma. But in this analysis, slightly different with the analysis done in establishing the reference interval, we still included the outliers here to further analyze the result of whether there is a significant difference or correlation between each group with the number of the blood parameters shown in the result. So, and here is the real statistical results in number here. As you can see that using the Kruskal-Wallis chi-square test with significant level 0.05 here, we concluded that the disease condition of birds only give a very slight response toward the the white blood cell level, or in other words, there is no significant change in either TCV and TPP and hemoglobin levels all of healthy birds or diseased birds involved in this study. So to further investigate on which groups that affected the white blood cell levels, we employed the Wilcoxon test with the same significant level or confidence level. It shows that only the birds contracted ectoparasite infection here, and the birds contracted with ectoparasite and pododermatitis here that could affect the white blood cell level. So however, there's a disclaimer, or it is worth noting that in this study, we actually ignored the degree of the pododermatitis and ectoparasite infestation in each individual. So finally, the conclusion of these studies are first to establish the blood reference interval of the Changeable Hawk-Eagle is quite challenging due to the limitation number of sample, and it is very challenging to create the subgroup based on the disease due to many limitations in the field in terms of diagnostic procedure, as well as the varying degree of severity at individual level. And then talking about the comparison study, that the significance difference among groups only occurred in white blood cell values between the healthy group and the group with ectoparasite infestation, as well as the group suffering from both ectoparasite plus the pododermatitis. So, and there was no indication of anemia in 3 disease group, and the results highlight the fact that actually inflammatory responses released to combat parasitism and parasitism with bumblefoot in this species are very much dominated by the dynamics of the white blood cell. So as a recommendation, this is pretty obvious that there must be more individuals involved in the study, especially in those that are considered as healthy individuals. This is especially important to establish the reference interval value, and it is suggested to involve individuals receiving similar husbandry management to prevent the appearance of confounding variables. So we would suggest that in terms to establish the blood reference interval value, the least different individual, it's better to use the individuals coming from the similar place with the similar rhythm of husbandry, like the feeding time, and then receive the similar condition or the cage situation. So yeah, there won't be more, or there won't be a confounding variables that may obscure the analysis. And this kind of study, we would highly encourage to be conducted and applied to any other wild animals, especially to explore more about the medicine or the clinical information of the wildlife, as we know that it's still very little, and it could be done actually in the form or using kind of retrospective study which use the existing or the past available data. So that's all my presentation. Thank you very much for the opportunity. Again, I would like to thank our supporter here, 2 universities, Gajah Mada University and Brawijaya University, for the funding and assistance for analysis, and Gembira Lokazu for the technical assistance, as well as the Ministry of Environment and Forestry of Indonesia for giving us the permit to conduct this research. Well, thank you.
Thank you so much, Dr. Irhamna. So yeah, we are quite ahead of time. So we have time for a few questions. If anybody has some burning questions to be asked.
I have one question for the trainer.
Yes, Prajalila.
Regarding the bumblefoot, I saw in your presentation you have 19 cases of bumblefoot.
Oh no, we have 19 unhealthy birds, but only around 8 individuals who were contracted.
Okay.
Bumblefoot, yeah.
All right. How severe the bumblefoot is?
Pardon?
Are you staging? What are the stage for the
Yeah.
bumblefoot?
Yes, that's our problem. We did not specify the grades of bumblefoot at individual, so that's one of our weakness in this study.
Okay, I don't mind about it, but just you know, bumblefoot can be because of the management problem, or you know, how you take care of the bumblefoot when the bird in captivity. Just concerned about it. Yeah.
Yeah, sure. I agree with that, Prof. Jalila, that actually we during this research we found bumblefoot with many stages, 1 until 4. So yeah, it's although it's very rarely the 4th grade, but we ever met one case for this bumblefoot. And
you familiar with ball bandage technique?
Yeah, but it would be happy for us to receive more experience from your side, Prof.
Okay.
Cases.
Can connect by email, okay?
Yeah.
To help with that, right?
Yeah, sure. Thank you.
Okay. Do we have questions? Other questions? So I'm sorry, I'm asking another question, but yeah, I was trying to interested to see if there were any sort of like risk factors associated with those animals that were considered the eagles that were considered unhealthy in terms of presence of ectoparasites and endoparasites. Were there any sort of like other sort of like factors that are present within the animal or around it that makes it more prone to having ectoparasites and endoparasites compared to the healthier ones?
Sorry again, if you may repeat your question.
Okay. So compared when you compare between the healthy and unhealthy according to your categories, so were there any sort of like risk factors observed in terms of management or the condition of the animals in terms of carriage of ectoparasites and endoparasites in one group? So what could be some of the factors that could increase the chances of those unhealthy animals to carry ectoparasites compared to the healthy ones?
Yeah. I think that's quite interesting question, Dr. Indah. That actually from the 33 individuals, actually they received quite a diverse health management, and from other, because we took them from different locations, right? But from our observation is that actually the condition of the cage, it may also impact or increase the probability of having ectoparasites, and also the endoparasites, especially because they always be in that cage for a long time, and the disinfectant probably it's also applied in several places, but not really every day or once a week. So I guess that could also increase the probability of getting the infection. And probably my colleague Bandang here, if you have if you have other thoughts about this, because he's also joining me in this research, you may also answer. Thank you.
Maybe suggest some statement that actually we have 3, right? 3 the birds with ectoparasite, and the location of the cage is quite nearby. So possibly maybe there's just one and it transmitted to the other one. But from when the ectoparasite comes, first we don't have any information about the record when we join this rescue center, so that the ectoparasite was already there before we joined this research center. The rescue center. So that we could not say where the origin of this ectoparasite, but we see that if the eagle was captured by the previous owner and the owner have many birds like the pigeon or something, and it can be transmitted by them actually, because that is trend in Indonesia that the collector always have many types of bird, especially in the protected one. So that's from me. Thank you.
Thank you. Okay, do we have any more questions? All right, if not, thank you so much, Dr. Irhamna, again for your wonderful presentation. And we would actually be going back to the first presenter because Mr. Sirawat Subanag from Kasetsat University, are you here?
Yes.
Okay, all right. So I would like to invite you to present your presentation entitled Prevalence of Avian Malaria in Raptors in Bangphra Wildlife Breeding Center, Thailand. So the screen is yours.
Okay. Can I share my screen right now?
Yes, yes, you may.
Okay. Do you see my screen now?
Yes, I can see your screen, but it's not in the presenter mode yet. Yeah.
Okay.
Okay. Yeah. Great. Yeah.
Okay. So let's start. Good afternoon, honorable participants. Today, I'm Cerebral, the veterinary medicine student from Kasetsat University, Kamphaeng San campus, Nakhon Pathom, Thailand. Today, we are going to talking about the significant disease in the wild raptors, especially in raptors. It's called the avian malaria. This project has the aim of the study is that because the center is responsible to rescue of both injured and confiscated raptors. So some raptors were being rehabilitated to be released in the future, and some of them are captured in this center and waiting for the protocol to be released. So therefore, the health checkups and disease investigation should be done to support the conservation strategies in Thailand. Before we going deep in about the avian malaria, I would like to introduce you to know this center. This center is located in Chonburi province in Thailand. As you see in the maps, it's on the east regions of Thailand, near Rayong province, and adjacent to the Bangphra Reservoir and caeruleus open zoo. And you see on this place, there also the sorry. Sorry, inconvenient. And here you can see our work that we work with the team from the Department of National Parks and Wildlife Conservation. So the works we collect the samples as a biometrics and blood sample to investigate the disease and help checking up. The wildlife that capture in this center, where are they come from? We have the team we call DNP Wild Hawk or Yeodong team. The Wild Hawk team will responsible into the working with the police, the Green Corps, to capture the illegally trading of wildlife in Thailand. And here you can see on the left, this one is one of the projects that they capture from the seller. This is a Changeable Hawk-Eagle, the nestling, and other wild birds also. And even there has a crested serpent eagle in this illegally trading. And here is the mission of the center. They have to rebreed some of the endangered species or invulnerable species. Also rehabilitated some of injured raptors, and also this one is a Brahmini kite that have training to be released soon. In each raptors, or most of raptors are captured in the cage that's appropriate habitat and have a suitable management during the period that they capture it and hospitalized. So now this is a major idea of our work. So this is the avian malaria is caused by blood parasite, which is called protozoa in Epicomplexa order, in the family of Hemosporidae. The significant genus is called Plasmodium species. We can see that in human malaria, you will see the Plasmodium vivax, but in the avian malaria, the significant species are in the 4 of them, including Plasmodium relictum, Plasmodium circumflexa, Plasmodium elongatum, and Plasmodium phalanx. These parasites are transmitted by the curiosity mosquitoes, such as Aedes or Culicoides, as you see in the picture below. And on the blood smear is a Plasmodium inside the red blood cell of the avians. And Plasmodium will make a significant illness in the wild birds, in juvenile birds, or a poor bird. So the clinical side that occur, we have investigated is on severe anemia that we monitor by the pack cell volume PCV and the weakness size of the birds. As you see in the research, prior research, we have found that Plasmodium circumflexum in a chikra, which is a juvenile chikra that migrate to Thailand and fallen. So we check up his health and we found that he got the Plasmodium circumflexum via the blood smear and PCR analysis. Another example of the sickness birds from avian malaria is from the Himalayan vulture, Gyps himalayensis. We have found these guys has a crop stasis and weakness signs. Until we check the PCV, we found that his PCV has dropped daily and weekly. So we decide to check about the avian malaria infection, and that's it. The avian malaria infection is persist, and we treat it with the doxycycline and chloroquine sulfate. to understand how the avian malaria is take a major role into the conservation of wild birds and raptors. I would like to introduce about this cycle. The cycle of avian malaria that has two cycles. One in the vectors is in mosquito, and another cycle is in the host. Firstly, the vector that taking the malaria sporocyte in the midgut here, the sporocyte will infect in the host cell via the blood suckling insects, and the sporocyte will go to the liver cells and transform itself to schizogony. It's called exoerythrocytic cycle inside the host. A lot of schizon has invade and multiply itself to merocyte. The merocytes will get infected in the red blood cell. We call this cycle is erythrocytic cycle. The merocytes transform itself to trophocyte. The trophocyte will undergo the cycle and replicate themselves severally until the red blood cell has ruptured. After the red blood cell ruptures, the gametocyte and schizon are mature enough to be infected in the mosquito again. Another mosquito ingests the blood milk inside them. The gametocytes exflagellation and fertilization in the mosquito, and then the cycle is completed into the oocyst and make a sporocyte again. So in this part, we see that if the hosts are unable to tolerate to the parasite, they will die. But some of the hosts can tolerate it. So the parasitemia will persist in the ecosystem. In material methods, we have to describe the sample collections and diagnostic technique. In this center, we collect in total of 13 species of raptors, including 8 species of Raptors comprising of rufous winged buzzard, black kite, Brahminy kite, crested serpent eagle, blithe hawk-eagle, changeable hawk-eagle, mountain hawk-eagle, and white-bellied sea eagle, as you see in the picture. And 5 species of nocturnal raptors, including barn owl, bar eagle owl, brown fish owl, buffy fish owl, and brown wood owl. The sample we collect the whole blood and EDTA blood from 74 raptors, which each of the sample we collect 0.5 to 1 milliliters, or less than 1% of their body weight. The complete blood count were performed, including packed cell volume, plasma protein, red blood cell counts, and white blood cell counts. The 4 parameters will reveal the health of the host via the concentration of the red blood cell, which taking a measure to transfer the oxygen in the bodies. And the white blood cells count will reveal the inflammation after the infection of the parasite. And to confirm the malaria infection, we use 2 techniques parallelly performed, which called blood smear and take a microscopic survey and molecular detection to confirm that is there any parasite DNA inside the blood. For microscopic detection, we use fresh or EDTA blood to make a smear. Sometimes, due to the hard working in the field, the fresh blood smear are lacked to preserve, so EDTA blood can use instead. And this blood smear will be dried overnight or maybe half a day and standing in with the right strain. The right strain is an in-house lab, which comprised of the right eosine methylene blue and Giemsa azure eosine methylene blue. This protocol, we follow the protocol from Germany. And the target cell that we need to see or we have to investigate is a hemozoin pigment. That is a byproduct after the hemoglobin degradation from parasites and which are the toxic to the cell. The parasite will produce the hemozoin pigment that is significant evidence for avian malaria. Another analysis is a molecular analysis where PCR, polymerase chain reaction. We use as the prior literature, they use the cytochrome B genes. That is a conserved area of the genome, which has less changeable sites of the genome. And the technique we use is a nested PCR. The primers, we decide to use 2 pairs of primers to determine is there any Hemosporidia protozoa infection in this bird with the Hem NF1 and Hem NR3 for the outer pair. And the second pair, we use Hem F and Hem R2. This will be reviewed on the agar gel electrophoresis at the site of the base pair at 500 base pair, as you see on the sample, and on the right is a positive control. After this procedure, to make a specific decision, we use the genus-specific primer according to Siloglu et al. 2019 protocol. For only positive samples on PMF, that's referred to Plasmodium-specific primers, and HMF, HMR, that's Hemoproteus-specific primers. As you see on the diagram, this will indicate that the size of the products from PCR that we consider to see. As a result, the disease prevalence of the avian malaria in this center we separate into 2 groups according to the activitiformis and strictiformis. For activitiformis, for the hawks, eagles, and vultures, we screened 56 individuals. Only 4 raptors reveals positive sample and positive to Plasmodium. In the 4 of them include 3 Nisaetus species, including Blythe Hawk-Eagle, Changeable Hawk-Eagle, and Mountain Hawk-Eagle, and 1 of Brahminicide. On strictiformis, we total screening for 18 individuals. Interestingly, the strictiformis are in fact 50% of the samples or of the population in the center. The most species has been infected are bar eagle owls for 6 individuals, and 1 of buffy fish owl and 2 brown fish owl. For the health impact, we demonstrate on the box and skewer plot. On the left is for accipitiformis, and on the right is from strictiformis. We declare on the negative samples and positive sample and compare together. We found that on accipitiformis, the PCVs of the positive and negative are 41% and 45.8%. On strictiformis, positive is from 36.89 and negative is 41.1. All the sample, there are no any significant difference between PCV, but as you see, the positive samples of strictiforms are significant different from the negative, the positive from accipitiform. This one is a clue of the avian malaria infection in different orders of raptors. So now we know that the avian malaria may not impact to adult raptors because we see on the PCV values, they are in the normal range that's from 30 to 45%. This consequence, as I revealed, I found that there has three hypotheses. One, the co-evolution of the host and parasite since the raptors and parasites are co-involute together. The second, the hosts are in the place where the avian malaria were persist. That means the immune system of the host will adapt to the vectors and adapt to the parasite that make them survive nowadays. The second point, avian malaria prevalence was notable on nocturnal raptors compared to diurnal raptors. This I have hypothesized that because of the nocturnal raptors are active in the nighttime, which directly to the vectors that the mosquito will go feeding ground on at night, while the diurnal raptors active on daylight, which has less vectors. And the habitat of these raptors are different because the nocturnal raptors are habitats in the hollow, in the woods, land, and swamp habitat, while the diurnal raptors are their habitat is on the high trees or less of the vectors. This involves the probability to the infection in these different type of raptors. If I have a comment for the further study, we will study on the genetic sequence of the Plasmodium clades to reveal the lineage of the infection species to analyze the behavior of infection in host species individually. We are also to thanks to the Bangphra Wildlife Breeding Center, Mr. Prasan Thongkong, Chief of the Bangphra Wildlife Breeding Center, and Ms. Chayanit Prasan Wong, the veterinary of Department of National Park, Wildlife and Plants Conservation. Thank you for your attention. If you have any question, feel free to ask. Thank you.
Thank you so much, Mr. Sirawit. So in the interest of time, if you have any questions, please feel free to use the chat box and you can contact Mr. Sirawit there, or you can also find his email address. I think you can put that as well in the chat box so that people can reach out to you. So thank you so much, Mrs. Rawit. That's interesting findings there about malaria in raptors. And so next up, we would like to welcome Professor Muhammad Izzat El-Zawalati, whom Dr. Puan managed to inform me that he is actually a UPM alumni back in 2012 to 2013. So currently, Professor El-Zawalati is based at the Uppsala University, and today he will be presenting about influenza A viruses among migratory Amur falcons and captive birds of prey. I would like to welcome Professor El-Zawalati. The screen is yours. Thank you.
Okay. Good morning, everybody. Thank you very much for the presentation and for the invitation. I think you see my screen, right?
Yes.
Okay, very good. So I would like to thank Professor Puan for the invitation, and today I will present our studies and research which we have done on influenza in migratory raptors, and this was a part of my surveillance project on avian pathogens in wild and domestic animals as evidence of zoonosis. Is a One Health research project. So the objectives today generally quickly will go through One Health and emerging diseases, and specifically virus zoonosis, and the research on virus threats using One Health, and then we'll focus on the detection of influenza in raptors, the focus of today's work. So virus infections, this is a global view, what we have been living for almost 2 years now, in a pandemic caused by a coronavirus, and still scientists and the public are searching for the origin of this virus. And this is a quick timeline of the newly emerging diseases, specifically virus infections. And as you can see, different viruses have caused different pandemics throughout the history. Down here is avian influenza viruses, and up is coronaviruses. We have 3 different coronaviruses, 2 very similar, the SARS-1 and 2, and MERS coronavirus from bats, and other viruses in mice and ticks. And these are wicked infectious disease problems, specifically affecting the food security and the food safety and antimicrobial resistance and emerging diseases. And while these are maybe interconnected, there is a strong interconnection. So how we can approach to these health issues? We can do this through the One Health research. We all living on this planet, and along with plants, other animals, livestock and wild. So there is a strong interconnection between the environment, humans, and animals. So we should consider, actually we have to consider an outbreak anywhere as an outbreak everywhere. As you may know, zoonotic diseases, almost 2/3 or up to 75% of the pathogens come from animals. So these called zoonotic, and they should have an intermediate host. And from this intermediate host, the virus or the pathogen can jump to humans, causing disease in a new host. And this may be due to some epidemiological factors, such as movement of birds and urbanization, close proximity, and land change, climate change. Other factors play a role in the emerging of zoonotic diseases. As we said, 75 up to 75 of diseases are from the animals. And this is called the One Health approach. For example, avian influenza or MERS or SARS or Ebola and other diseases in companion animals and other hosts. This is a live animal market. Imagine in the United States where different animals are grouped together. And this is a nice wild, maybe one pathogen can be the next pandemic agent. So how these pathogens are generated within the animals. So the virus is generated or is present in the reservoir in the wild, and then it can jump either through an intermediate host or can jump directly to infect another host and then causing the zoonotic disease or the pandemic. And this is called the zoonotic disease pathway from the animal to the human. And there is another way, anthropozoonotic disease, or the reverse zoonosis, where humans can infect the animals again. This is a model, a very interesting model, how the viruses generate to cause a pathogenesis or virus adaptation, and then can infect. As we mentioned, this is the objective of our project, which is a One Health project. This is an example on the Rift Valley fever, how this virus typically represents a One Health approach. So how we can approach this? We either do research in the wild animals or in the humans, but what is interconnect between both? It's the One Health. I am emphasizing on this presentation on One Health for the sake of the importance nowadays with One Health and zoonotic diseases. These are images from Africa, and this is from Southeast Asia, where these humans go in the wild and collect animals and contract a new disease and then go home, spread the disease between other people, and then the pandemic starts. So there are different projects already established on the search for pandemic threats, like Preempt. This is a military research project, and Predict and other Virome project. So the animal health matter then, because we have almost 2 million cases of it may cause deaths. So there are a number of 13 zoonotic diseases can cause this. This show you the interconnection between animal, human, and environmental health, and how this is very important. So there is no one discipline can track this. It should be interconnected or multi-sectorial or multidisciplinary or interdisciplinary. There should be some sort of cooperation between all these sectors to tackle the disease. And this is called the One Health approach. And this is a new educational program. Like, I was very happy to see the One Health Malaysia has been established in UPM. And I am aware of this in IPS. So the One Health, as we mentioned, is an integrated approach to improve the health of humans on this earth. And there are different global changes that favor the emergence of infectious diseases. As we mentioned, the increased toxicity or the other ecological factors, biodiversity or climate change, and different industrialization and travel. These are different factors that cause the emergence of new diseases. This is a nice summary of the different important respiratory viruses that has affects humans through a number of years since 1989. And we can see influenza and SARS are causing issues. And this, where we study this One Health projects, we study them either in the field, in the wild, or the markets, the farms, or where animals and humans and environment interconnect. And as I mentioned, different important viruses such as influenza, Zika, dengue, and we focus on coronaviruses. We focus on respiratory viruses in this project. So wild animals are very important to as a very important part of the One Health strategy. However, limited studies has been found on wild raptors, specifically, as we will come through. So imagine one person can cause 0 case, can cause different disease in different people. So viruses are a global infectious challenge, specifically influenza viruses, and viruses don't recognize borders. A virus infection or outbreak in somewhere can easily transmitted through travel. You can see the heavy traffic of travel busiest day from one place to another. So viruses goes viral, and imagine you are traveling on a plane, and then this plane has been quarantined because there is some sort of threat or pandemic threat on that plane. This is real scenarios of this is a flight from Dubai to New York, where the few travelers has been infected with influenza. So these are updated map of the preparing for the next pandemic, new emerging disease or re-emerging or deliberately emerging disease. And this is the different patterns of zoonotic diseases worldwide. So we have to focus on different areas where disease can be generated. And again, this is showing you how the disease from one place can easily transmit to another place very quickly. This is called the spillover, or the zoonotic spillover, where the virus transmits from the wild life to the humans. And specifically viruses are very important attention. So what is the next pandemic of a bird flu? We don't know yet. So we have recently been affected with different viruses like corona, typically corona SARS-CoV-2, and this is in 2019, and very recent a new porcine coronavirus in Haiti, and another porcine canine coronavirus in Malaysia. I'm working with this group in Duke University. So my research group in South Africa and USA and Sweden, we work on microbiology and virology research group. We focus on emerging infectious disease and influenza. Specifically, we study the micro and macroecology, as well as we study antimicrobial resistance. And we hypothesize that there is some sort of distribution and prevalence of these viruses or pathogens in the wild and in the environment may typically affect humans. And we do characterization of zoonotic antimicrobial resistance in live food chain. So in my research group, we focus on whatever new agents or new viruses, as well as the commonly known viruses, such as influenza and coronaviruses. And also we use a One Health approach. We use different methodologies, molecular, phenotypic, until whole genome sequencing, and as well as serological methods, and we study these agents. This is part of my studies in USA, where I do the avian influenza surveillance in the United States. So a true picture of the emerging disease will only exist if we conduct a programmed surveillance effort. This is a general flow chart of the methodology we follow in my lab, where we do sample collection, and then we go ahead and do molecular characterization, real-time PCR, and we follow a number of steps until subtyping. Then we go for full characterization by sequencing to detect the agent. We can also do virus isolation. So for our focus on the species today in raptors, I conducted this study for the first time in the world. As you can see, this is the origin of avian influenza in China, in Southeast Asia. So from this location, the virus can spread to North America, to Africa, to Europe, and it's already has spread worldwide. And it's a typical agent of One Health. And these are the migratory flyways where the birds, migratory birds, can move from one place to another throughout the whole universe. And this is a influenza transmission. So we conducted these studies in South Africa. This was a large project including all type of birds, because there was a very limited data on influenza in South Africa. So I conducted this study on different wild birds. We go in the field and collect the samples from the wild. For example, this study has been was published in 2019 with Erasmus Medical Center, where we collected birds from about 550 birds from 270 species, different species, different birds, and we found influenza in a very low percentage. However, this is an evidence that there is a prevalence of low pathogenic avian influenza in South Africa. And these are our photos in the field where we collect the samples. And this is our photo in the Raptor Center, our focus today, where I go and collect the samples. This is Ben Hoffman, very thanks for him. He was very cooperative in during the sampling of the raptors. And these are our field trips during sampling of Amur Falcons. This is myself carrying the Amur Falcon with Rina Pretorius from South Africa. And this is, we go to the field and collect the samples very quickly to the lab with different cooperation from different people. So for the detection of influenza in raptors, when I was in South Africa, there was a number of outbreaks of avian influenza, and the exact origin of these viruses, how it come to the domestic poultry, is not yet known. So I was in a discussion one day, and then I was informed that there is a number of raptors coming down from northern China and Mongolia throughout until South Africa. And then this for the wintering area, where the weather here in South Africa during the winter, it's very nice. So they come from the breeding area down to the wintering area. They do the, so when it is winter there in the Northern Hemisphere, they come down to South Africa, better climate. So they migrate all over this place, distance, and stop in a city in South Africa called Newcastle. So before that, there was no study conducted on to screen influenza in migratory Amur falcons. However, the previous studies on different species has been reported the presence of Newcastle and low pathogenic and high pathogenic influenza viruses in other countries, but not in South Africa. So, and also there is a very limited information on influenza and Newcastle in avian and other avian species in South Africa. So I planned this project. We went to Newcastle and we captured the Amur falcons. You can see here the number of birds, very huge number of Amur falcons. And this is a city of Newcastle. And these are the Amur falcons on the trees here in South Africa. So we plan to detect influenza A viruses. And this was another parallel project on trapping of these Amur falcons and follow up these using transmitters. So a group from Germany with Rina Pretorius, they were putting the tracking device on the falcons. So while they are tracking the raptors, we collect the samples side by side, and then we screen them for RNA. Actually, this project is very laborious and they require a huge cooperation of effort and very costly. As I mentioned, this is the first study in the world to detect avian influenza in migratory falcons or migratory Amur Falcons. So after RNA extraction, we follow the same methodology which I have previously mentioned, and then we do a number of tests on these samples. We also did sequencing and virus isolation. And by the way, this is the location of KwaZulu-Natal, and this is the location of Newcastle, and this is another location for other captive birds, captive raptors, because also we planned a wider study, not only from the migratory falcons. We want to see the whole picture of raptors in the country. So we collect from another places. And this is the tracking locations. This is a nice photo for the raptors, as you know, Amur Falcons. So the results here was very exciting. So we detect influenza. We collect samples from 9 species of birds of prey in South Africa, and these are captures. And here we can see that we collect from African goshawk, Amur Falcons, from 12 birds, owls and peregrine falcons, sparrowhawk and the snake eagle, yellow bell kite. So this was a very nice cohort of samples. And then we collect from each bird 2 samples, one oropharyngeal, another cloacal, and then we do the test. So we collect, and then we do the molecular test. So we detect influenza in 14 samples out of 48. Total number of samples from different locations. It's a high percentage of positive result in this study, where about 14 samples, which represents almost 45% of the samples. And then here we collect the samples from these birds, and then we move to other also birds. So we collect the samples from other birds. And this study has been published already in transboundary emerging disease. And luckily we detect 1 sample in positive to be positive for H5. So after we detect the influenza, we test for subtyping H5 or H9 in these samples, and then we do subtyping, which subtype of virus. So we were able to detect H5 in 1 Amur Falcon. However, we tried to isolate different viruses other than influenza. So we detected Newcastle. We didn't find the Newcastle. And we also tried virus isolation and in embryonated chicken eggs. All this study has been published already, and then you can go ahead and check them. So from this study, we have to be prepared to the next pandemic. What is the next pandemic will be, although we are currently in a pandemic due to another virus. So we have to be prepared, because an emerging disease like viruses can cause a lot of challenges and trouble in the human life. And we have to be prepared specifically for influenza. There is a lot of influenza cases now reported in humans from avian type in China. And what is the next disease? We still don't know. And for this, I would like to thank the grant supporters. This study was supported by the National Institute of Health, United States, and Center of Excellence for Influenza Research and Surveillance, and colleagues in St. Jude Children's Hospital, Duke University, Erasmus, and very thankful to the World Working Group of Birds of Prey in Germany. And without this, we were able to, with this, we were able to study the influenza in Amur Falcons. And this study has been very nice and very interesting, adding information to the literature. And with this, I hope I am on time, and thank you very much.
Thank you very much, Professor Zewalati. That's such an impressive work that you have done, and eliciting the importance of considering One Health in helping the world to be better prepared should the next pandemic come, So yeah. If there are any questions for Professor Zewalati and the rest of the speakers, you can continue to interact in the chat box, because we will have to move to our last but not least presenter from Taiwan. So I would like to welcome Associate Professor Hui Yun Seng from the National Taiwan University, Taipei, Taiwan, to present her work on short-term exposure of anticoagulant rodenticides enhances the toxin accumulation in prey to predator. So the screen is yours. I would like to welcome.
Okay, thank you. Thank you for the introduction. Hi, thank you for attending my talk today. My name is Hui Yun Zeng from the Raptor Research Group of Taiwan and National Taiwan University. So on the behavior of our research team, I want to share a study about the toxic accumulation in the body of prey and predators during the 4-year exposure of anticoagulant rodenticides. So as you know, the most famous case of pesticide overuse is DDT, and it also marks the dramatic population decline of both eagle and osprey. So here we want to talk about the effect of rodenticides on non-target animals. The rodenticides are used to control rodent populations, and rodent control is important in Asian countries, especially for the countries that agriculture is the major financial support. And the first generation anticoagulant rodenticides, FGARs, such as warfarins, they were widely used in most countries since 1950s. However, because of the long-term and unlimited use of FGARs, more and more rodents have developed resistance to the rodenticides. So the efficacy of the rodenticides started to decrease. And therefore, in 1970s, more toxic and longer half-life rodenticides were developed, and they were called second-generation anticoagulant rodenticides, SGARs. Such as bupivacaine and difenacan, and sooner become the most widely used rodenticides among the world. And many studies reported the secondary poisoning of non-target animals by anticoagulant rodenticides, especially raptors. Because raptors, many raptors, they are opportunistic predators, or they are scavengers. So they can capture less active or dead animals poisoned by ARs easily. And in Taiwan, the first generation anticoagulant rodenticides were used since 1950s, but due to the resistance to FGARs, it was replaced with SGARs since 1980s. And in order to reduce the agricultural economic cost, the government provides SGARs to farmers for free during October and November every year from 1980 to 2015. So here you can see here the important studies show the high frequent detection of AR in different raptor species. These species uncover the serious situation of secondary poisoning of raptors in Taiwan. So in this study, we want to focus on the black-winged kites, Elanus caeruleus. The population of black-winged kites have expanded. Before 1990s, the species mainly distributed in Asia and Europe and Africa, and then they expand into Europe. So from the data collected from eBay, we can see the expansion of black-winged kite from 1990s to 2010. And the figure on the right-hand side shows a dramatic population increase of black-winged kite in Taiwan. This species was discovered in 1998 and then continuous increase within 20 years. And here's the, So here's the diet composition of breadwing kites listed here. And it shows that mammals are their major prey. And you can see that rodents, more than 90% of prey number and biomass are rodents. And other prey, such as the red-collared dove and the barred button quails, are also found in their diet. Because the major prey of breadwing kites is rodent, so therefore we want to know whether breadwing kites accumulate higher concentration of anticoagulant rodenticides than other species. And this study not just focus on the raptor, we also want to know their how the situation of their prey. We want to know whether their prey can survive longer after eating rodenticides. So the first aim of this study is to quantify the accumulated rodenticide residues of the kite. And secondary, we want to construct toxicity assessment of rodenticide exposure in their prey, Rattus losea. So here, totally 112 individual gunshot and bird strike black-winged kites were collected from 7 airports. And these airports were located in the Western Taiwan. And the red dots indicate the location of the airport on the map. So the breadwing kites, they prefer to inhabit in open areas. So the airports are these kinds of habitat. But because of the flight safety, the airport needs to remove the breadwing kite by gunshot. Then these individuals were used to examine the rodenticide residue in our study. And most previous study used individuals from rescue centers or using caucus to examine the residue concentration of ARs. But our study using healthy individuals from airports. And this study was conducted from 2013 to 2016. So the entire liver, the experimental process is here. The entire liver was storage in 75% ethanol in dark condition at -20°C refrigerated. And the AR residue was analyzed in the component and followed by usual procedure. And the 7 toxicities of ARs were examined, and including 3 compounds of FGARs and 4 compounds of SGARs. In the following analysis, but we only focus on the major rodenticide bodifocum. In this study, we also separate all the samples into 2 groups, the subadult and adult. Subadults are individuals younger than 2 years old. So the young breadwing kites have the rufous crown and the breast, and the white fringes on their back and greater covers. The adult looks like this. They have very white breast. So here's the second experiment. This experiment is the toxicity exposure trial of the rodent, and totally the 106 individuals were collected from the wild, and we choose individuals with 220 grams of body weight to decrease the individual variation. And all the rodents were collected also from 2013 to 2016. And before the trial, the rodents were kept in a cage for 1 week individually, and then we will not provide food for 1 day before the trial. And 50-gram bags containing 0.005% bodifocum and water were provided. So during the trial, 24 hours later, the bags consumed were weighted, and then we will provide normal food to the rodent and record the survival period until the rodent died. So here's the result. We compare the morphological trait of black-winged kite during 4 years. And the characters, including the body mass divided by tarsus, and also the wing cord and the tail length. The numbers in the blanket are the number of our sample size. So there's no significant differences among years. And here's the figure, shows the changes of detection frequency and the concentration of bodifocum during the 4 years. And the blue bar indicates the proportion of individuals that contain bodifocant. And the green bars indicate the proportion of individuals with detected residue above 0.1 ppm. And the yellow line here represent the average concentration of bodifocant, and the red line represent the maximal concentration among all the individuals. So both the detection frequency and residue concentration shows the increased trend. The proportion of samples with residue above 0.5 ppm become tripled from 20 to 60, and the yellow line shows the average concentration doubled from 0.1 to 0.2. And this figure shows a similar trend in adults and subadults. The residue in the livers of adults and subadults increased during the 4 years. So here's the result of the second experiment. After feeding bats with a bodifocant, the average survival days increased during the 4 years. So in this figure, we can see that the average survival days is 4.5 in 2013, but increased to 7.9 in 2016. And this table shows that, and this table also found that the percentage of individuals survive longer than 8 days. And the maximum survival day also increased during the 4 years. So here's this table shows the frequency of detection and residue concentration retained in the body of diurnal raptor species. And this is including the kestrel red-tailed hawk and red hawk, and also the black-winged kite we show here in red. And some species such as the red-tailed hawk and black-winged kite they have very high detection frequency, and the kestrel and red kite, black-winged kites, they have relatively high residue concentration. So compared to the nocturnal raptor species, only greyhound owls, they showed the high detection frequency. But the mean concentration among all the listed species, black-winged kite have the highest concentration in this study. In the previous slide, we mentioned the important study conducted by Dr. Hong, and this study showed the frequency detection of anticoagulant rodenticides in raptors in Taiwan. And as you can see here, there are 5 species. This is black-winged kite and black kite, crested goshawk, collared scops owl, and crested serpent eagles. Among these 5 species, the black-winged kites in detection frequency or concentration, they have they are the highest one. So while the black-winged kite have high residue concentration of anticoagulant rodenticides, we propose that the diet and the life history trait may enhance the accumulation of anticoagulant rodenticide. According to previous study, we can roughly estimate the number of prey hunted by black-winged kite in a year, at least 1 rodent per day. So totally there will be 365 rodents per year. And during the breeding season, there will be more rodents captured to fill a fledgling. And black-winged kites have some special life history traits that can be very affected the accumulation of AR, such as they can breed when 1 year after hatching, and they can breed twice annually. And also, if a pair of kites, they could raise 3 to 4 fledglings in a single breeding attempt. So because the anticoagulant rodenticides can affect non-target animals, so we suggest the use of rodenticide should be replaced with biological control, such as attracting raptors to the areas with serious rodent damage. And the use of artificial perches in the agricultural area can efficiently attract the raptors to perch and hunt. Here's the artificial perch here. And here are some images from the trail camera, which was set on the artificial perch. And many raptors use this perch to hunt, and it might be an eco-friendly method to control rodent. So here's the conclusion. The frequency of detection and concentration of Brody-Falcon in breadwing kites increased from 2013 to 2016. And the survival period of Rattus losea, their prey during after feeding Brody-Falcon, significantly increased during the 4 years. And the use of rodenticides should be replaced by, with biological control, which not only reduce the economic cost of rodenticide, but also reduce the secondary poisoning of non-target species. In this study, we want to thank Wu Xuelu and the members of Taichung Wildlife Rescue Group for assisting in collecting the samples, and also thank other organizations for their suggestions and support. Thank you for your listening.
Thank you very much. Are there any questions for Dr. Hu Yun? Thank you so much for your sharing your findings. I'm pretty sure it would be very useful, adding to the body of knowledge in terms of helping the efforts of conservation and raptors, and also in improving One Health throughout the world globally. So if there is no question, thank you so much. And we are actually at the end of the concurrent session 7. Thank you very much to all speakers and to all of the participants attending this session. I would like to invite all of you back to the main room for the closing ceremony that is happening now. Thank you so much again. Appreciate all your attendance.